Welcome, everybody. Thanks for joining us for the, this must be the 5th of these environmental science talks that we've been hosting over Zoom. And this evening, we're going to talk about hooligan ecology, history, and cultural significance. We've got Meredith Pochardt, who used to be here with us at the Watershed Council and now works for NOAA Fisheries, and Ted Hart, fisheries specialist at Chilkoot Indian Association. And the research project that we've been working together for a long time is based at the Chilkoot Indian Association, and we're going to be doing that again this year and continuing on for hopefully into the future. So with that, I'll introduce Meredith Pochardt and Ted Hart.
Cool. Hi, everyone. So Ted and I haven't actually rehearsed this together, but Ted will take the first couple slides. As Derek mentioned, we're basically going to talk about the hooligan population study, and I'll go into full screen mode actually. So here's the kind of overview of what we'll talk about today, but Ted will start us off with some of the background on Eulachon, and maybe I should start off by saying there's a lot of different a lot of pronunciations. Eulachon, hooligan, and then the Tlingit name is saak. And so any of those might be said, but they all mean the same little fish. We'll talk about the background and the traditional uses of hooligan, the population monitoring project, and some of the new methods that we've been using, environmental DNA, and then some of the challenges, and then what we're looking forward to moving forward. So Ted, I'll just move the slides and just tell me when you want me to move them.
Sounds good. Thanks, Meredith. So I'm Ted Hart, the fisheries specialist at Chilkoot Indian Association. And Chilkoot Indian Association started this project in 2010. And it was kind of in response to the southern distinct population really having noticeable downtrend, and even near extinction in some of their runs down there. So you can look at the map here, and so California, Washington, Oregon, like southern BC maybe. It's all the southern distinct population. And what we see up where we're at is the northern distinct population. So we wanted to start monitoring our run to see if we noticed any differences in the run size. There is some concern from elders and locals that we're seeing less up here. So we kind of wanted to get somewhat of a handle and kind of get a population estimate each season to see if we notice changes or some longer trends. So then we began doing that in 2010, and we hope to keep doing this each season. So the next slide. So Tlingit, it's a very important little fish up and down the coast. They're known as the salvation fish. And I believe that has a lot to do with their timing. This time of year, all the winter food supply is, you'd imagine would be nearly depleted by this time or already depleted. And then all of a sudden, you get this huge return of these very nutrient-rich little fish, and with them, brings marine mammals. That's food also, larger fish, birds. So really important little fish, and you can imagine with a name like salvation fish, we really want to keep these little fish around for many generations to come. So, next slide. This is a picture of the process of making hooligan oil, or in Tlingit, it's saak hooligan oil. So this is what really makes these fish very prized, is their fat content and our ability to turn them into very nutrient rich fish oil. And you know it's done by catching large amounts and basically letting it ripen for they say maybe 4 to 10 days, depending on the weather or your personal preference. So you let it ripen till the desired amount that you'd like, and then you take large amounts into these great big cooking pots of water, and then you boil that, get that water nice and hot, kind of break the fish down, stir it around a little bit with like maybe an old paddle or something, and then you can start pouring. So there's a great big fire underneath this good big cooking pot right here, and then towards the end you can just skim all this beautiful golden oil just right off the top of the water. And some people, you know, they might get maybe like 5 gallons of oil just from one cook, and then you can also do multiple cookings. So they're very high fat content. And you know where we're at now, De Shoop Haynes is the end of the trail, and that being part of the grease trail that was almost very reliant on the trade of hooligan oil to our neighbors up in the north. It's you know, you get up here in the northern regions, and these lipids and fats, they're basically currency up here. It's like liquid gold, they say. So it makes a lot of sense that you know, that would be the currency. So very, very important little fish that we want to keep around. Next, please. Yeah, so here's the process of skimming the oil off the top. You can see the old paddle there. It was used to break up the hooligans. The fermenting process that helps break them down, and you can get more oil that way. And also, this is a condiment, a medicine. You know, all types of cure for humanity, all types of things. This oil. So you can just skim it right off the top like that. Dwayne Wilson. This is out at their family camp out at 4 Mile, and yes, their family has been cooking oil for many generations in this very spot, right on the river. You know, in previous years, the saak, the Eulachon, they would be swimming right by the bank right here, and you could just scoop them out with the dip net, transfer it right into the ripening pit, and then into that great big cooking pot right there. You can see there's a little chimney, and so the fire would be lit directly underneath that pot. And they just, you know, you can cook it all day long. And he's got his paddle there to break up the hooligans, and those guys have made some really, really high quality oil right there for many years. You can just see it in Dwayne. He just really loves doing it. Next, please. All right. Lonnie Hotch, you know, very amazing woman, subsistence food guru, and you can just see it on her face, you know, how happy she is to have that saak liquid gold right there. So just one tablespoon is enough to sustain our elders, you know, for an entire day. They're highly nutritious. It has a very long shelf life. You can preserve things in it. It's it's very, very healthy for you. Take a spoonful every day, and you're going to be a pretty healthy individual. And yeah, it just really brings a smile to people's faces. And yeah, we want to keep those smiles coming like that because of the hooligan oil. All right, so this was referencing the southern distinct population listed as threatened in 2010, and it's not a coincidence that that was when our study started. So we wanted to try and get a handle on our population size and see, try and track any kind of trends, any kind of things we want to document, and just try and keep an eye on this important species in our northern population. So our first method that we started doing is the marker capture. So we wait until the saak, the Eulachon, start entering our river in the Chilkoot River. That's been pretty consistent for us. It's easier to trap in that river. We put up these spiky traps, and they swim right along the shore, and we put up these wings that kind of guides them right into our traps. They kind of create a little bit of an eddy, a little kind of some softer water for the Eulachon to swim into. And once they swim into there, then we count each fish. We count every single one that we catch. So we have probably a group of 5 counting, dipping, and marking. And our trap is right above the river, or right above the bridge, the Chilkoot Bridge. So we catch, count, and mark. So and we give each one a little clip. We call it the shark fin clip. So we remove just a portion of the adipose fin, and then we release the fish right back into the river, just right there. And then we mix them in with all the rest that we don't mark. So we're only marking just a portion of the run. And then those are released to mix in with the fish that are unmarked. And after a few days of counting and clipping, then we go up and we do recovery. So we look for these shark fin clips, and we do that up above the weir, anywhere from the weir on up to the lake, usually. So it's just catching many more Eulachon, saak that are marked and unmarked. And it takes a while to find a marked fish. It's kind of like a needle in a haystack sometimes. And another good way to really get some good numbers and count is to take a subsistence user's catch. So we can just go through, hey, do you want help, carrying your catch up the bank? Do you mind if we count these real quick and look for marks? So we just count and go through thousands and thousands. And if it really feels like you won the lottery a little bit when you get the marked fish, and you hold it up, hey, I got a marked fish, and verify it with your partner over there. And so that's how we get our estimate for the marker capture. We mark, let them mix with the unmarked fish, and then we look for them again, and we're able to come up with our estimate each season for the marker capture. And it works pretty well. We've seen a big variation in numbers and we've been able to document almost each year an estimate out of the Chilkoot, and it's been really good. It's been a really good river for us. Really good location, and we're able to get some good data out of there.
Yeah, I think there's a couple of photos here.
Cool.
Yeah, I don't know what the slide is, but
yeah, so this is talking about our monitoring efforts, and yeah, you know, there's questions, you know, does it vary much on year to year? And with our estimates, it has varied quite a bit. So here's some pictures. You know, sometimes, you know, there might be a little snow on the ground, little spring storms, but yeah, that's a picture of our trap just above the bridge there, and we scoop them out. Try to do we try not to get too crazy with our sampling. You know, we try and give them a little space in our little troughs here. We give each one water, and we try and release it back into the river as quickly as we can. And so clipping each one, counting each one, we end up handling quite a bit of these little guys, but that's kind of what gives us our good data in the end, is try and get as many numbers as we can. And we've got little clickers, little tallywhackers. You know, we're counting each one, trying to make a nice uniform clip each time, so they'll be easy to recognize upstream. Yeah, some people like to get our own little techniques and, you know, styles for, you know, doing the sampling. It can, you know, it's a lot of time. Just it can be cold, but yeah, I'm really grateful for our workers out there getting these numbers for us. There's some more pictures. This is more of the recovery portion. So in the top picture here, you can see the weir down in the background. So these guys are all above the weir, looking for fish that are mixed in with the clipped and unclipped fish. So again, we're counting each one, and then looking for those clipped ones, and each one goes right back into the river. And if we find a clipped one, we'll give it a little secondary mark, usually on the dorsal fin. So we don't count that one twice. But yeah, it can take quite a bit to get a few clipped ones up there, and sometimes you'll go forever and you won't get anything, and then all of a sudden you'll get a few back to back, and it always kind of ends up evening itself out in the numbers. Like the more you get, kind of, yeah, we just try and get as many numbers as we can, basically. So this is how we get our estimation each season. You can see, let's see here. So it can vary quite a bit. Let's see here. So 2018, you can see we marked and counted 30,000, and then we counted 18,000 for the recapture, and then we ended up catching 64 marked above the weir. And then that equaled out to
sorry.
That's right.
Not on that. Sorry, I can go back.
Nearly 9 million, 8.7 million for that year's estimate. So we go through that same process each year, and it can vary a lot in our estimates, and also our captured, or the ones that we've counted in the the pens. So yeah, it's just got that's a formula here, and we type in our numbers, and that's what gives us our estimation for each season. And yeah, it's really important to keep a good track of our numbers. That's a lot of numbers, so that's kind of the biggest part of the game. It's just keeping good numbers, keeping good data, and then that'll end up giving us a pretty good estimate for that season. This is a little bit easier to see our final estimates. So yeah, lots of variations in our estimates each year. 2019, that was definitely one to remember. We came in at like, it's at 26.8 million for our estimate, and that was doubled our previous record. So that was pretty amazing to see. Definitely what we saw, big numbers, and it was consistent, and it sustained for a really long time. So yeah, I feel really good about these estimates that we do each season, and we can get to see it firsthand, and so we can look back and remember the size of the run, and actually be able to put a number to it. It feels pretty good. And if this, of course, this isn't a perfect way of getting an estimate. It has its flaws, but we definitely are able to get a pretty good handle on our estimates. And one of the challenges are everyone else that's out there trying to subsist on these guys. So sometimes we might be marking a bunch of fish, and some subsistence users are above us, most likely catching a bunch of our marked fish up there. We can imagine, but that's just part of it. They have every right to be there, and it's been going on for many generations. And also, we've pretty much had to focus on the Chilkoot, and all the while the Chilkat is a huge run, and we just haven't been able to get really good numbers over there because just the size of the river and how braided it is. It's really hard to predict where they're going to go each year. They can be on this side of the valley, or they can be all the way on the other side. They might go way up the river. They might stay down in the estuary. And also, we have to hire a pretty large crew to get these numbers for our market capture. So it's also very costly, and could be about 20,000 annually for 5 to 10 day season. So it's definitely got its challenges to be able to sustain into the future, but we've been doing pretty good so far. We hope to keep on going and keep getting these good estimates into the future. And I'll let Meredith take it from here. We've been real fortunate. She's been our eDNA guru, and this feels really cool to be using this kind of technology, and to see the pretty decent results that I'll let Meredith tell you guys about.
Yeah. Well, thank you so much, Ted. That was good. Ted and I gave this presentation a while ago, but we haven't necessarily rehearsed it again. So it was good that you just jumped in and took it. It was awesome. Yeah. And And the mark recapture method is like that's kind of a tried and true method for developing a population estimate for a lot of different species. And, it's kind of the go-to method. This eDNA or environmental DNA is relatively new. It's becoming much more popular, but it's still new. And yeah, so it's one that we started piloting basically in 2014 just to see if it would work. And yeah, it seems to really have been working. So we'll go over some of the data, but basically the background on like what is this eDNA stuff is it's picking up DNA fragments that are just in the environment naturally. So people are getting them from soil samples. We're taking water samples and finding hooligan DNA in that. There's also some folks doing it with just air samples and looking at aerosols. And so it's this kind of inclusive term that refers to sampling DNA from the environment rather than the organism itself. As I mentioned, it's a pretty new tool and it relies on this mitochondrial genome, but don't worry too much about that. And so one of the first papers that talked about using eDNA was back in 2008, although a lot of the fundamentals of these methods have been around for a long time, but using it for like wildlife management applications is relatively new. And then some other papers and studies came trickling in, and now like if you were to Google search eDNA studies, you would find like hundreds or thousands of them out there. It's a new technique that has been picking up a lot of speed lately. I guess we will talk a little bit about the mitochondrial genome, but a little bit of old high school biology. We have a bunch of mitochondria in our cell that has DNA as well as your nucleus. But there's many more copies of mitochondrial DNA within each cell. And it's also a shorter sequence. So it's easier to be able to detect it with different types of equipment. And so, and also your mitochondrial genome, the mitochondrial genome is mapped for a lot more species than the nuclear one. And so this is, just kind of walking through how we collect these samples. We collect this as sampling out of the Katsahine River, actually, and we just scoop a bag of water. You filter it down. This is one of our field crews in Berners Bay, filtering it down. And I'm not sure if you guys can see my mouse or not, but those are the filter cups. And then within that is a little filter that collects all the DNA. And so that is collecting all of the cells that are in that sample. So there's probably hooligan DNA. There's maybe also some out-migrating salmon smolt. Maybe some dollies are swimming around. Maybe definitely gulls and marine mammals are around. And so it's collecting all of that DNA onto that filter. And then we take that filter, we partner with Oregon State University, and they do this lab work there, but they are able to extract all the DNA from that sample. And then we use this other process called ddPCR to look for just the hooligan DNA. And so we've gone through and collected the sample, filtered it down, extracted all the DNA, and now we're looking just for how much hooligan DNA is in that sample. And I guess, in the age of COVID, we've probably heard a lot about PCR sampling. This is a method. A lot of COVID samples are slightly different, but this is one that we're using that makes it really easy to quantify. And I don't want to get too into the weeds on this, but it is pretty cool. So basically that sample with all the DNA in it, it partitions that sample into like 20,000 droplets, so tons of little bubbles. And then if there's hooligan DNA within one of those bubbles, it will turn positive. And so you basically just count the number of positive droplets, and that's your DNA concentration. And so you'll see that DNA concentration used a lot in some of the other stuff, future slides. So this is an example. This is our like positive control that we run in the lab. And so this one has like an actual piece of hooligan tissue in it. And so everything is positive, has so much hooligan DNA in it. And so you can see here that all of those little bubbles are blue and they're positive. It's a lot of hooligan DNA. when a sample is really rare, maybe they're just entering the river, maybe it's a really small run, we'll see fewer positive droplets. And then if they're really abundant, this is a sample from one of the big years on the Chilkoot. And so this is what it looks like when, there's tons of hooligan in the river and there's tons of positive droplets, but there's also quite a bit of negative ones that didn't. So not every droplet is turning positive, but a lot of them are. So that's kind of how that works. There's a lot of things that affect the concentration of DNA that will be in the sample. Different species shed DNA at different rates. Also, different individuals can, especially at different times in their life. If you think of like a salmon, like a coho in the ocean versus one spawning, obviously a lot more DNA is being shed while they're spawning. And then there's a lot of other factors that can affect DNA, the sunlight or temperature. And so one of the things that we do when we take our samples is we're, trying to keep them in coolers, get them filtered and stable as soon as we can so that we don't, and that DNA is not degrading. And then some of the other factors that might influence your DNA concentration is like how much water is being transported downstream and basically diluting the sample. And so we do flow correction on rivers where we have stream discharge to kind of correct for that. But that is a challenging thing in this area where a lot of the rivers that we're looking at just don't have stream gauges. So these are some of the results, and this is I don't want to get too into the weeds on these graphs because they're a little tricky to read, but this is looking just at the Chilkoot River, and they're broken out into 3 different panels. So in this first one, we have 2014, 15, and 16. Those were lower returns. And then this middle one, we have 17, 18, 19. That was like kind of our medium return. And then our 2020 return was way bigger than even what we saw previously. So we're still analyzing results from 2020. And so, but it looked like 2020 was a huge year for DNA. And then with these results, because as Ted mentioned, with the marker capture, we kind of plug all of our numbers into an equation and we get one estimate. And so, that's how it comes up with this one number. And you get a standard error of, this margin of error on either side of that, but you get a point estimate. And so with DNA, you're not necessarily getting that. We're taking samples daily throughout the run and just monitoring that concentration of DNA throughout the run. And so you're seeing these concentrations obviously are fluctuating throughout the run. And so the metrics that we can use to somehow get a population estimate out of this DNA concentration that's constantly changing, you can look at the overall just size of the peak, like how big is that peak? And is that an indication of how big that run was that year, or the whole area under the curve? Because there is basically this curve, and a lot of these, some years are kind of multimodal, or there's a couple different peaks that are happening throughout. And so those are the 2 metrics that we're using to gauge this DNA concentration and use it for a population estimate. And here. And so this is looking, comparing it. Again, all of this data is from the Chilkoot. And so this is comparing it to the marker capture data. And so these 14, 15, 16 were relatively lower years. And our eDNA concentrations also ended up showing lower concentrations. And then 17, 18, and 19 were higher years, and our DNA concentrations were also higher. We don't have marker capture data for 2020 due to COVID last year, but we were able to collect eDNA samples. And so we can't pinpoint this in a comparison with a marker capture estimate for last year, but last year was a big run. I think 2019 was also a really big run too, so I don't know if it was that much bigger than 2019. But yeah, it falls like it would been nice if 2020 fell like in the middle of our span rather than like at a far end. It's hard to pinpoint what that estimate was. And then this is comparing that. And so this is looking at that size of the peak and the area under the curve again. And this data has been log transformed, but it's comparing it to the marker capture estimate in millions. And this is actually doesn't include 2019's big year in this, but you're able to see that kind of really nice one-to-one relationship that we're getting from the eDNA data, comparing it with the marker capture data. Like when we have big marker capture years, we're also seeing more DNA, and vice versa. When there's a lower marker capture estimate, we're seeing lower quantities of DNA. Again, 2020 is an outlier on this, and yeah, it falls off these off this graph. And then, so that was just comparing the Chilkoot data, which we started doing eDNA sampling on the Chilkoot in 2014. And then in 2017, through some additional funding, we were able to bring in more of a regional approach. And so then we started looking at Eulachon runs in Berners Bay, looking at the Berners River, the Lace River, and the Antler River. And then also the Katsahine, the Chilkat, the Ferriby, and the Taya and the Skagway rivers. And so we were able to look at this broad range and see what was going on with these populations kind of on a regional level. And so because a lot of these rivers don't have stream gauges, we're not really able to correct for that kind of flow dilution factor that could be happening. And so these are more comparisons for that river from year to year rather than comparing it to a different river. As of now, that's kind of what we're conservatively using these for. We're also looking at the peak of these concentrations versus their latitude. So basically like north to south gradient, or south to north. Because some folks might hear even, like hooligans spawn much earlier in Ketchikan on the Unic River, and then they kind of are marching their way northward, it seems. There's obviously like some different populations worked in there, but it is this kind of, I don't know if we've coined the term or not, but been calling it the grease wave, similar to the grease trail, but that this just as an ecological resource, these hooligan are, it's a continuous resource. And so you have this, they're not all of the rivers are spawning all at once, but they're kind of happening in a progression. And so for marine mammals, they're allowed to take advantage of a lot more of the resource because they can kind of follow it as it's progressing. And then, yeah, looking at regional trends, there's definitely been some interesting things that we've seen throughout the last few years of the regional study, where one river will be, usually is a small run, but then just goes gangbusters. And then other ones that might be like bigger are having lower returns. And so there's definitely some regional fluctuations that are happening. And so this is just a snapshot of 4 of the rivers. I didn't want to make a super crazy graph figure, and so I just chose 4. And this looks at that 2017 through 2020 eDNA concentrations at these rivers. And you can see, obviously, they're different levels. The Ferriby seems to have a much higher concentration in a lot of years. Definitely in 2019, the Ferriby had a huge spike of DNA. The Katsahine slightly less, but there's also a lot more water coming out of the Katsahine, and so that dilution factor could play a role there. But you're seeing that both the Ferriby and the Katsahine had pretty big runs in 2019. And then you come down to the Chilkat, and that green line was very small in 2019 on the Chilkat. 2017 was a bit bigger there that we're seeing. Again, these Chilkat samples they're all from Jones Point. And so the way that river's so braided, it's hard to get a true representation of what really is going on. So it's just sampling basically one braid, but just noticeably overall across the entire river, the Chilkoot was much lower in 2019 than a lot of the other rivers were. And then looking at the Taya River, you can see it's kind of like a lot of them are really similar. Definitely 2019 and 2020 were pretty similar size, and also the timing was very similar. So it's kind of interesting to look at. And so this is talking more about that kind of latitudinal migration and the grease wave, and kind of prolonging that resource for marine mammals and seabirds and just the ecological resource. And so this is just looking, there was only in 2017 and 2018, those are the only 2 years that we had in Berners Bay. And so they're the only 2 years that we have data from that kind of lower latitude. But we were able to see that there definitely is this kind of latitudinal migration where they were peaking in Berners Bay, and then, progressively moving their way northward. So yeah, some of the kind of the take homes. The EDNA has been a cost-effective tool, and it definitely allows us to look at the rivers on a broader range. But I would say also that the Chilkoot marker capture data and its ability to confirm what we're seeing with the EDNA is also very important. And I think that's like one of the things going through the data, and it's like, whoa, 2020 was a huge year, but we didn't have marker capture data to compare that with. And that, there's always some, always wish that you could do more. As far for EDNA, it's definitely the approach for being able to monitor, several different rivers within a region. It would be, just the Chilkoot is it sets itself up really nicely for a marker capture study, but most of the other rivers in the region are they're too braided and silty and too much flow, and it would be too difficult to put a marker capture crew on the ground there. So some of the things we're looking forward to in the future, these are these auto sampler units, and so it's kind of in this cooler here. These are being developed by the Opens Lab at Oregon State University, and this cooler will be able to basically sit anchored on shore. You'll run a hose to the river where you'll secure it to a spot in the river where you'll want it to sample from, and then it will be able to continuously sample you can take 24 samples. And so that will definitely improve our ability to sample at a broader range. One of the things in 2017 and 2018, when we had a crew in Berners Bay, it was awesome, but it was also a lot to man a crew to go out there. And they were out there for like a month at a time. We wanted to make sure that they were there before the run happened. They stayed throughout the run, and then we want to get a couple samples usually as that's kind of winding down. And so, it does mean, camping out at Berners Bay for a month, but some springs are really awesome and some springs are a little wet. And then looking kind of on a regional level and being able to branch out to other tribes that are interested, that have hooligan runs and want to, implement some monitoring efforts to look at how their trends are doing. And then, there's definitely, just aside from the population aspects of hooligan, there's a lot of other unanswered questions. We don't really know how old they are when they spawn. We don't really know if they spawn more than once necessarily. There's a lot of different theories out there. And so, they're definitely an understudied fish in a lot of aspects. But, so here's some of the different life history traits that have been found is in the Columbia River, that they spawn about when they're 2 years old. In Alaska, between 4 and 5 years old. And then, as I mentioned, there's discrepancy between if they spawn more than once or just once, like a salmon would, and then die after spawning. If folks have have been, it's definitely kind of assumed that they die after spawning, but definitely not like the way salmon are, where you can see their carcasses, all over the stream. You definitely see some washed up alongside the rivers during spawning, but not the full mass of what is in the river. And so, yeah, that's still kind of a question out there. And then, yeah, as Ted talked about, there's that northern distinct population or southern distinct population that's Washington, Oregon, California, that's been listed as threatened. And then there's the northern population, which our rivers are part of. But then even within that northern population, there's some different population structures. And just understanding that can help you understand the health of those populations a bit better. And yeah, with that, I mean, this project has been going on for quite a while. We started in 2010, and there's a lot of people to thank. Definitely all the partners on it and the funding organizations and all of anyone who served on a crew. It's definitely a lot of work.
Right on. Thank you, guys. Thanks, Meredith. Thanks, Ted. And yeah, let's take some questions and have a discussion. We've got some in the chat here already. I'll try to start with the first one. How do you decide when to start recapture? And that would be a factor in how many are recaptured. Yes?
Yeah. Ted, you want that
one? We like to give them a little bit of time to get established. So maybe a good part of the day, we might see them showing up early in the morning, and then we may, towards the end of the day, be counting and marking. And a reason for that is just kind of letting them get established a little bit. There are a lot of beliefs that we should give them time, even subsistence fishing. Really give them a day, even a couple days, get established, and that'll really ensure that they're going to have a big return for that year. That season's going to be a nice long return. So I like to give them a little bit of time to get going.
The next question's from Dina Selby. The number of hooligan is way up in 2019. Does that mean whatever is being done, limiting fishing, is a success?
There hasn't really been necessarily like a limit on fishing. I think there's most likely a lot of factors that are at play, but yeah, I mean, one of the 2019 Chilkoot data is really high, but 2019 Chilkat data was very low. And that's something that we saw, I forget off the top of my head which year we had a really low Chilkoot year, but we found that they're very sensitive. And so these are only two occasions where this has happened. So yeah, 2015 was a very low Chilkoot year. It was also the year that they were pile driving for the ferry terminal. And I know there's no scientific evidence for this, so I'm just going off observations, but they're pile driving during the run. And folks that are familiar with the area, that you have to go, fish have to swim by the ferry terminal to get to the Chilkoot River. Saw a very low return on the Chilkoot that year. Similarly, but all the other rivers were doing really well. And then in 2019, there was work happening on the highway, and blasting was happening, and that was along the Chilkat River. And so 2019 Chilkat returns were very low, but every other river was very high. Those are observations. There's no scientific evidence, but it's happened twice. So.
And those observations do correlate with traditional knowledge too, right, Ted?
Yeah, absolutely. There are many taboos involved with the saak harvesting, many more than any other type of subsistence harvesting, like salmon or anything. So yeah, just giving them time to get established when they're first coming in, not disturbing them, throwing rocks, things like this, and just giving them respect. And it's even said that a long time ago we would put away our differences, that we had in the past, and we all would need to come together and just have good intentions while we're down there, good, and not fighting with one another kind of thing. And even that makes a difference in return for the saak. So yeah, they're very prolific, but they're also very sensitive too. So yeah, it's definitely traditionally that's been said for a while.
The next question, also from Dina, is: do the runs cycle like salmon? Say hooligan run is better every 3 years. How many years do the hooligans stay out in the ocean?
Those are still unknown. We don't necessarily know how old they are, but most information points to that they're 4 to 5 years old, and so you could think that the cycles might be 4 to 5 year cycles. But I think, yeah, there's a lot of other factors that are at play also, in how big these runs are and in which rivers.
I think you can almost correlate the big salmon returns too with big Eulachon returns. They definitely come in and they clean the gravel, and they create lots of food from the ground up, from all the little microorganisms up. So a big Eulachon return usually means that in 4 years or so there's going to also be a big salmon return, hopefully.
It's harder to detect in hooligan that, like with pink salmon that are coming back to their natal stream every time, you can really see that signal of that every other year cycle. But it appears that hooligan may not do that. A hooligan that was born in the Chilkoot may come back and spawn in the Ferriby, or so we think. So it'd be harder to detect that in data, that real strong, every 3 year cycle, say, because they're not coming back to the same system, or they may not be. We don't know for sure. Clay asked a question: how old are these fish when they spawn? I think we talked about that a little bit already. Rustin says, I just started working for the Forest Service in Wrangell this week. The Stikine has no data on Eulachon, and I would love to try and spearhead a monitoring program. There's a large subsistence harvest, according to the people I've talked to.
Yeah, we should talk, Rustin. I think it'd be great to get some data on the Stikine.
I would love it too.
I haven't heard of any work going on down there. Has there anything happening out of Wrangell or Petersburg?
No, I mean, yeah, I think that there is none. There's been some limited studies on the eunuch, but I haven't heard of any on the Stikine either. So
yeah, all the people that I've talked to up here said that they don't know what's going on. They just know that there's a harvest, and they'd like to know. So yeah.
But yeah, we should get in touch.
The 2020 numbers are so high and anomalous. How confident are you in them? That question's from Paul Cunningham.
Yeah, I think we got to do some more digging still, but I mean that's what the data is. But yeah, they're very high.
Well, we also changed the Chilkat sampling last year. We did something completely different in 2020 that we had never done before, which was, one, we took samples from a skiff right off the mouth of the Chilkat. We did a transective sampling, and we also hiked across the river a couple of times, although that didn't really factor into those graphs probably. But so we did a few different things in 2020 that may have impacted the Chilkat numbers anyway. The Chilkoot, though, is still
very high.
Very high. Yeah.
Yeah, and the Chilkat graph is just using the Chilkat graph that I presented. It was just the Jones Point data. So it didn't incorporate those other sampling locations that we added in last year. Just for continuity, I just used the same location. So Yeah, 2020 was a really big run though, too. So it was big. Was it double our previous big run? I don't know, but yeah.
Pam Randall's asked about the range of dates for the arrival, or of the run, probably.
Yeah, so I looked that up today because I was also kind of curious on it. But the earliest we've seen them is the 20th of April to show up, and or I guess I should say the earliest that we've started the mark recapture cruise is the 20th of April. And so, as Ted mentioned, we might have seen some fish in the river the day before that, but wanted to let those scouts kind of get established. And so 19th, 20th of April has been the earliest. And then the latest that we've seen them arrive, that we've started our cruise, have been May 6th. And so that's quite the range.
Anybody else have questions? You can raise your hand or just speak up. Oh, there's another one. Pam Randall says thank you.
Thank you, Pam. Yeah, Rustin, I'll throw my email in the chat, and then you can shoot me an email if you want about try to figure something out for Forest Service monitoring.
Yeah, absolutely. I don't know what my email address is yet with the Forest Service since I just started, but I'll throw you tomorrow an email from my account and we'll figure something out.
Yeah, thanks. That'd be awesome.
Yep, and we'll be out there again this year taking eDNA samples. Probably not mark recapture again, but yeah, you'll see us out there.
Yeah.
All right. Well, thank you everybody for attending, and thank you very much to Ted Hart and Meredith Pochardt for putting together a presentation and talking about hooligan. And today it feels like spring is here, and maybe the fish are going to be here in a few days, and the birds are here, and you know, it feels pretty good to have some sun and some warmth.
Yeah, it's very exciting.
All right. Well, thank you guys, and everybody have a good evening.
Yeah, thank you all. Thanks for
Everyone.